I'd now like to introduce our next speaker, Professor Ian Goodfellow. Ian is Professor of Virology and Deputy Head of Department of Pathology. A few years ago, during the Ebola outbreak in West Africa, Ian led UK volunteers in setting up a diagnostic laboratory at a treatment center in Makini. He recruited and trained local people, making it possible for them to turn samples around in 24 hours and quadrupled the country's testing and diagnostic capacity. Ian subsequently collaborated with the University of Makini to establish a research facility for post-Ebola capacity for endemic and potentially new and emerging viruses. Ian is going to tell us about his efforts in West Africa now.
Thank you. So good afternoon, everyone. I should start with just a very, very brief introduction. I'm a basic scientist, so I'm trained in molecular biology and biochemistry, and 95% of all the work we do in my laboratory has nothing to do with anything I'm going to talk to you about today. So we study noroviruses and gastroenteritis, and all this work has really come about from a desire to act as a volunteer during the Ebola virus response. So I'm going to talk specifically about our capacity building and engagement activities that have come around since the tail end of 2014. And as this slide really needs not very much of an introduction, but just to remind you that the Ebola virus epidemic that we experienced between 2013 and 2016 in West Africa was the worst that we've seen to date. It affected 3 countries, Sierra Leone, Guinea, and Liberia, and there was in excess of 28,000 cases, with 11,000 deaths. And my laboratory, myself, and a number of people from Cambridge got involved in this in various aspects. I was initially there to set up a diagnostic laboratory in the northern districts in Makeni. This was at the tail end of 2014. And then subsequently, we set up a genomic sequencing facility in the Ebola treatment center so that we could provide real-time genomic support to the epidemic. I would like to highlight that I am not the only person from Cambridge who's been out to Sierra Leone and contributed. I may have been one of the first, but there's a list of individuals from various parts of the university who played various roles. So people from my laboratory were involved in diagnostics and sequencing, people from the Department of Medicine, including from Andrew Lever's laboratory, from Public Health England, the hospital, also from the Sanger Centre, and these guys played a very important role in our research, and also from the MRC. So a huge range of people from Cambridge deployed as volunteers to provide laboratory support during the epidemic. And as Watu indicated, I led a team back in November, December 2014 to open up one of the first diagnostic laboratories in the northern districts of Sierra Leone. And this laboratory operated until 2014 to September 2015, testing somewhere in the region of 15,000 samples, and played an important role in testing of samples from the northern areas in Sierra Leone. But in March, April 2015, we had the idea of putting a genetic sequencer in the tent next to the diagnostic laboratory. The idea being that we would be able to sequence Ebola viruses as soon as we could get the samples from the patients. And this enabled us to understand virus evolution, and more importantly, at the tail end of the epidemic, gave us an additional piece of information for contact tracing and identifying the sources of infections and characterizing cryptic chains of transmission. So as you can see, we brought in quite a lot of equipment. We were funded by the Wellcome Trust to the tune of around £400,000 to fly in all this sophisticated equipment to essentially take a piece of DNA and sequence it in its entirety in this tent. And I don't want to really go through the research in any great detail, but not surprisingly, we published a few papers on this. We primarily used the sequencer, I think, to greatest effect during the tail end of the epidemic, and I'll talk about one example later in my presentation, where we would see individuals appear in areas of the country where Ebola hadn't been present for many months, and we would be able to identify potential sources of infection and rule out certain hypotheses. This video just shows an overview of all the genome sequences that were collected throughout the epidemic. This outbreak was in fact one of the best sequenced of all infectious disease epidemics, with somewhere in the region of 5% of all cases sequenced. And this has enabled us to perform some quite detailed analysis on factors that contribute to the spread and sustained transmission of Ebola. This is published here. I just highlight quickly that over a third of all the genomes that were published as a result during the course of the epidemic came from our tent, and with the help of Raoul, who is here in the audience, who is a researcher from the University of McKinney. Now, near the tail end of the epidemic, so in and around July 2015, the epidemic was winding down, and it became clear to us that the treatment center was going to be closed. So we had invested all this money, time, and effort to get all this infrastructure in place, and we were going to be left without a home in September of 2015. And this is when we met the Vice Chancellor of the University of McKinney, who's here, who's also joined us today, by Father Joe Turre, if you want to wave. And we had a discussion about what could we do with all this infrastructure. Could we donate it to a local university? Could we establish a new research facility? And this is where the idea of setting up the University of McKinney Infectious Disease Research Laboratory came about. And they provided us with a very nice building. This is was in fact a law clinic, and we were able to then move all our equipment into this building to enable us to maintain the sequencing capacity. As you might imagine, there's a bit of structural changes and a few things we had to do to the building to get it ready. This is July 2015. We shipped over some biosafety cabinets. Some of these purchased off eBay, some of them donated by various companies, some of them borrowed from other laboratories that were in country and then closing down. We shipped everything in. We had some benches made by local craftsmen at the side of the road. We actually had to ship in the tops of the benches because it's a very specialist sort of material, and we couldn't find that in country. And the laboratory actually opened in September 2015. So in September 2015, the Bishop and the Minister of Health opened the facility, and we were there ready to provide capacity training, capacity development, and also sequencing support, because there was still ongoing chains of transmission in and around the border districts of Sierra Leone at that time. Now, this is a picture of the laboratory in September 2015. Here's Raoul, here's Luke, and another local scientist that we had trained in Ebola virus sequencing. And it's fair to say it's a pretty sophisticated lab. We can do full genome sequencing. We can do immunoassays. We have luminometers. We have 2 biocontainment suites. And it became very clear to us that establishing this capacity was very, very important, and maintaining it in the future was also important. And this was really highlighted in January 2016. So in January 2016, on the 15th, the World Health Organization declared the epidemic over. On the same day, I was walking in to work, and I get a phone call to say there's been a new positive case. So this rings a huge number of alarm bells, and lots of questions were raised. So the questions were, was this a new introduction? Where did this virus come from? There was a real worry that this virus was part of an unknown chain of transmission that had been incubating somewhere in a remote village, and we would all of a sudden go from 1 case to 50 to 100 or something. Or was it exposure from a survivor or a new zoonosis? At that time, it's 15th of January, I'd gone home. All the international scientists had pretty much left. So the only people who were available to do this were the local scientists. And luckily, Raoul and Ja were able to take this sample within a period of 24 hours, sequence the genome of this virus, and then we were able to rule out many of these hypotheses. We were able to figure out that, in fact, the closest known viral sequence we had in our database was from 2014, so almost 2 years from the date it was collected. And this told us quite clearly that this was not an ongoing chain of transmission. This wasn't a new introduction. And in fact, this new case had appeared via contact from a persistently infected survivor. So the laboratory is still operational, and in fact, it's starting to take on a life of itself. I need to be clear and say that this is a University of McKinney facility. This is not a University of Cambridge facility. We are there purely to help support them. And there are other researchers who now use the facility for a number of research projects, some of which are listed here. Probably the largest research project that is ongoing within the facility is funded by USAID and is run by UC Davis. This is the PREDICT program. And they seek to try to identify the potential sources of spillover events. They are sampling 9,000 domestic and wild animals and screening them for highly virulent viruses, including Ebola. But we've also provided support to the Ebovac vaccination trial, the TKM siRNA trial, an ongoing nutritional trial. And there are a number of other researchers that work in the facility and pay UniMac for the access to the facilities. Now, to move slightly different angle, I'm going to talk about the public engagement work that we've got involved with more recently. And this really came about from discussions that we had with the local population and local people we were working with in the treatment center. And it's very clear that there are a huge number of factors that contributed to the size and the rate of expansion of this epidemic. One of which was the fact that the local communities really don't understand what infectious diseases are. They don't really know what a virus is. There's a lot of superstition, a lot of rumors. These are some of the rumors that were pushed around at that time, and probably the worst being that the healthcare centers were essentially centers for harvesting of organs. But all this contributed to the lack of engagement with the response teams. So we were left with the question, how can we improve science education and engagement in Sierra Leone? And we met this guy, Andrew Ben, who is a hygienist, but actually his day job was a science teacher. And of course, he was made unemployed as a result of the Ebola outbreak. He was playing the role of a hygienist. And together we came up with the idea of running a public engagement program in Sierra Leone to try to make high school students better aware of the infectious diseases they're exposed to in their everyday lives. So we got some funding from the Wellcome Trust, and on the 21st of June 2016, we held a one-day workshop here in Cambridge, which was organized by Cambridge Infectious Diseases. We brought together a bunch of people who were like-minded and had an interest in public engagement, and we came up with some ideas. On the 21st of August, we then flew out, along with all this equipment and reagents and stuff that we had to use. We employed a number of local science teachers and then started to deliver this public engagement program. And the program's been running since 2016. It is a partnership between us here at the University of Cambridge, the University of McKinney, and a local charity known as Educate. And essentially we go from school to school every week, and we teach over 4 days about infectious diseases and the various, and I'll just run you through some of the various activities that we go through. So on day one, we really focus on trying to give the students an idea of what are infectious diseases, what are parasites, what are bacteria, what are viruses. We get them to make models. This is primarily aimed at senior secondary school students, but the age range is anywhere from 15 up to 28, depending on when they start school. Also, as part of day one, we give the students access to microscopes. We get them to look at water samples that we've collected from the local environment, including what they think is drinking water. One of the most popular activities is that we're able to generate a laser-powered microscope simply using a handheld laser pointer like this. And if you shine this at the right angle through a drop of water, you can magnify parasites and bacteria to such a degree that you can actually see them on the wall. And they find this quite amazing that they can take a sample of even bottled water, and if you get the angle just about right, you can see all this stuff swimming around in it. And this is on day two, we talk about disease transmission and prevention. And one of the first activities we do as part of day two is we covertly infect somebody in the classroom, and their job over the rest of the day is to infect as many people as possible. So halfway through the day, we do a big reveal and everybody finds out they've got UV gel all over their face and on their arms. And this is really just to highlight how fast pathogens transmit from person to person. We give the students an opportunity to dress up in PPE, and we talk about what's the function of different forms of protective equipment, and we do some hands-on activities that kind of model routes of transmission to give them an idea of how fast pathogens can spread in an epidemic. Also, as part of the last day, this is very popular, we stole this idea from Northampton General Hospital. This is a YouTube video. We teach some good hand washing technique to, this is Gangnam style. I'm sure many of you know this. On day 3, we focus on the immune system. We talk about what our immune systems are, how they function, why we don't die every time we get infected with any pathogen. We show them various videos of different cells from our immune system. This is one of my favorites. I think this is a neutrophil chasing after a bacteria until it finally catches it and eats it. This is a macrophage doing largely the same thing. We have some mocked up experiments. These are rubber pathogens. We run in a sort of an assay where we get them to do a mocked up ELISA to try to figure out what somebody has been infected with using millets with iron filings and rubber pathogens, some of which have got magnets in. The final day really focuses on epidemiology. We've developed a board game which illustrates how vaccination can be used to prevent transmission. And we give them the experience of working as a sort of time-traveling epidemiologist. They take on the role of Jon Snow, and we give them various bits of information over the day, and they've got to figure out where the source of this cholera outbreak is. So just to run through these very quickly, this is the amount of students we've engaged so far, 1,600 students. We also run teacher training workshops where we try to talk more specifically about general science topics. This is run by Ian Harvey. So our overall goal is to make McKenney a centre of excellence for science and research, and also to provide the local communities with the knowledge to help prevent future outbreaks of diseases occurring again. Lots of challenges. I think everybody here has highlighted all these challenges about sustainability, and there's also a real lack of homegrown expertise. But with the University of McKenney, we're now running an undergraduate training program where we run undergraduates as volunteers through the research laboratory to train them in molecular methods. I'll just finish by thanking everybody involved in the work, and again thanking the team from the University of McKenney. So Raoul, Father Joe and Mercy are here today for providing support over the last few years and enable us to establish this research facility. Thank you.
Thank you, Ian. Another presentation packed with a lot of capacity building efforts. We're going to take 2 or 3 questions, but please keep them very brief because we don't want to be out of time. There's one question right here. Over here.
Sorry, I just wanted to add quickly. Concerning the various myths of origin of Ebola, could you please, as a professional in the field, tell us the actual or the real origin of Ebola? And could you please tell us as well if scientists have found a cure for it, or is still a case of HIV and AIDS that people are still waiting for the cure, a breakthrough in science? Thank you.
Okay, well, thanks for the question. In terms of the origin of Ebola, I think that's a kind of contentious question. There's a lot of people looking at this. The dogma is that it is bats, and you can find bits of Ebola virus sequences in bats. You can experimentally infect bats. But as far as I'm aware, no one has actually isolated live Ebola virus from a bat yet, although the UC Davis team may or may not have managed to achieve that. But I think the indications are that bats are probably the natural host. In terms of a cure for HIV, I think it's clear to say, in my opinion anyway, is that, if you have access to good healthcare and antiretrovirals, you can live a relatively normal life. But the problem is really getting access to that, sustained access and monitoring over the duration of your illness. So I think we've gone a huge way in terms of our ability to treat HIV. The problem is really getting those drugs to the people who need them the most, and also the underlying infrastructure, so be able to do CD4 counts, monitoring drug resistance. That sort of infrastructure is really what we lack now, rather than actually having the drugs to treat.